I'm going to be doing those spectacles. Thank you everyone for coming along this afternoon to our Honours and Masters talks. It's really nice to actually have a bunch of people here in the room with us, as well as those who we hope are listening in online. I'd like to begin with an acknowledgement to country. Today we are meeting on Lutruwita, Tasmania, Aboriginal land, sea and waterways. I acknowledge with deep respect the traditional owners of this land, the Mirina people, on which we meet today. I pay my respects to elders past and present, and to the many Aboriginal people that do not make elder status, and to the Tasmanian Aboriginal community that continue to care for country. I recognise a history of truth which acknowledges the impacts of invasion and colonisation upon Aboriginal people, resulting in the forcible removals from their lands. I stand for a future that profoundly respects and acknowledges Aboriginal perspectives, culture, language and history, and a continued effort to fight for Aboriginal justice and rights, paving the way for a strong future. As I said, thank you to our students. There's a bunch of staff. There's nice to see some support from other postgraduate and other students in the room and our industry partners. And I don't know if we have other guests and family and friends online as well. Thank you all for coming along. This is really important because it marks the end of the research effort for our Honours and Masters students. And it's really not just their research projects, but this is the pinnacle and the capstone of their entire studies with us. So we're all very excited to hear what you've been up to over the last little while. Just a little bit of housekeeping for people who are here with us. In the unlikely event of an emergency, please exit out there and muster over the road in the car park or on the other side of the road where the oval is. If you need to leave to go to something else, if you can restrict yourself to doing that between talks, that would be much appreciated by our speakers. Audience members online, who I assume we have some online, please note that this is being run as a webinar, so you have been automatically muted or videos turned off. To ask questions, can you please type with the Q&A function? And when we get to the question section, someone will be checking in on those questions and will present them if they're not asked by someone in the room. We have two Honours students talking today. They have 15 minutes for their presentation. Our first speaker is a Bachelor of Agricultural Science student, Nikita Ng, who's going to be talking to us about blueberry rust. Nikita, would you like to come up and tell us about your project, please?
Good afternoon, everyone. Thanks for being here today. And as mentioned by Shane, we're going to be presenting about breaking the disease cycle of blueberry rust today. So for starters, I wanted to talk about the blueberry industry in Australia, which was actually valued at $400 million in 2021, and it is still currently rapidly expanding with conventional and organic production systems all across Australia. Blueberry rust was detected in Tasmania in 2016 and was then considered endemic since last year. And the causal pathogen of this disease is Thekopsora minima, or the blueberry rust pathogen. And it is a biotrophic fungal pathogen that relies on living plant tissue to acquire nutrients to survive. The main host of this pathogen is in the Ericaceae family, and important crops that are infected by this disease is, of course, blueberries, which belong in the Vaccinium genus. And the symptoms of this disease include the purple patches, as you can see on the top image, and they will appear on the upper leaf side, as well as the visible raised yellow to orange pustules, which are the Uridinea spores on the underside of the leaf. And they will usually also have chlorosis and necrotic spots that turn brown and eventually cause premature leaf drop. This is a typical rust fungi life cycle, which displays the 4 main spore stages of the rust pathogen. And in that red circle there is the main issue of eradication in Tasmania, as only the Uridina spores have been detected so far. The Uridina spores are abundantly produced in that Uridina spore life cycle, and that can happen for an indefinite period of time, which is why it can spread to other crops, other blueberry crops indefinitely. The 4 main control forms for blueberry rust is chemical control, which involves the use of approved fungicides that have been known to reduce disease severity or incidence, and they're listed on the top of the slide. And the second one being cultivar tolerance, where it is also a good option to use tolerant cultivars of blueberries that can withstand rust infection. And the third one is a very important one, which is cultural control, involving the maintenance of hygiene in farms, regular disinfection of used equipment, tools, and anything that's come into contact with the plant, basically, as well as disposing infected plant materials so that the disease doesn't spread even further. There's also defoliation for non-deciduous varieties, which removes the plant part on which the spores usually overwinter. And there are also some potential biological control forms, but they have not been discovered yet so far, so we won't get into that. There's also the research objectives for this project. First one being, would defoliation be a viable method of management for breaking the disease cycle of blueberry rust for semi-deciduous varieties of blueberries? And then we have which organically approved chemical defoliants or other defoliating agents can be used under the Tasmanian climatic condition. And then there's also, does complete defoliation affect the fruit yield and quality of the plants that are treated? And the final one being, how does temperature conditions affect the Uridina spore germination? So this project was separated into 2 parts, the first part being the defoliation trials to trial the defoliating agents. And then we have the second part, which is the spore viability trials, where we will try and incubate the spores and observe their germination. So the first part of the trial, we had 7 different treatments, the first one being control to compare with all the other treatments. And then the second one is defoliation by hand. And the remaining 5 treatments are just the chemical defoliants that have been known to cause leaf senescence at high rates. So the first chemical defoliant that we used is called Protone. It uses abscisic acid as a main ingredient to induce the expression of senescence-associated genes. And the second one being VBC, which has 1-aminocyclopropane carboxylic acid as a main ingredient. And that is also the direct precursor to ethylene in ethylene biosynthesis, which have been known to cause leaf senescence and abscission as well. And zinc sulfate is known to be a micronutrient that is typically applied through foliar application. And when it is applied at high rates, it can cause localized toxicity on the leaves, which then causes the leaf to drop, I guess. We did assessments of the plants. We did pre and post-treatment counts to observe the effects of the defoliating treatments to the plant leaves. And we did the new leaf growth and pre-existing leaf counts at the same time to see if the plants were able to recover from defoliation. And we did flower counts as well to assess potential yield production of the plants that have been defoliated. So after the berries were ready to be harvested, we also wanted to assess the yield to see the effects of the defoliation treatments on the yield. So we harvested the blueberries and then weighed the yield of fruit that was produced by each individual plant. And we also did individual fruit weight as well as total soluble sugars, which we measured from the juice of the berries. Moving on to the results, we have the effects of the defoliation treatments on the number of leaves. As you can see on the zinc sulfate ones, we did see a significantly reduced number of leaves by about 90% on average. And then on the hand defoliation one, which is this one in the middle, as you can see, it was 100% because obviously they were done by hand. The VBC and protein treatments we didn't include in the results or any further assessments because they didn't seem to cause the plants to experience any notable amount of leaf drop. The second part of the results were the new leaf growth. This was, again, to observe the recovery of the plants. As you can see, the hand defoliated ones did have a considerable amount of new leaves on them at this point in time, although the zinc sulfate ones were not significantly different from the control ones in terms of new leaf growth. So with the flower numbers, we did observe that there was 70% less number of flowers in the zinc sulfate plants, while the hand defoliated ones did not seem to be affected by the treatment in terms of the number of flowers, as they had a similar number to the control plants. We also have the results on the yield produced by the plants. And as you can see, the results for the zinc sulfate ones, they were reduced by 50% on average for the yield. So the hand defoliated ones produced a similar amount of yield to the control ones. So obviously hand defoliation did not have a significant effect on the fruit yield. And then on the individual fruit size and weight, we observed that there was no significant effect from the treatments on the size, on the weight of the fruit on average. Although from visual observation, we did see that on the zinc sulfate plants, they were largely varying in size within a single plant, which is pretty noticeable. And then we also have the results for the measurements for the total soluble sugars or total soluble solids. We did see that in zinc sulfate berries, they did tend to have a higher total soluble sugars, which is around like 1 Brix units more than the control and hand defoliated ones. But the hand defoliated ones did produce fruit of a similar sugar content to the control plants. And then moving on to the second part of the project, we have the spore viability trials. We conducted 2 initial trials before the start of the project. The first one was an attempt to produce a bulk inoculum of spores, just so that we can have access to fresh spores that were not contaminated by external contaminants, which sometimes happens to the field samples that we get. And then we also did an agar screening to decide on what type of agar we were going to use to inoculate the spores onto. And we did water agar, which is just plain water agar, which is used for a lot of fungal inoculum typically. And then we have blueberry leaf extract agar, which is our attempt to mimic the growing conditions on blueberry leaves in a natural environment. And then we also did an antibiotic agar that was also an attempt to isolate the spores, but no difference was observed. So we didn't continue with those plates and we just used water agar plates. And according to existing literature, we did find out that down to 15 degrees Celsius, the spores were still able to germinate in a previous research. So we instead used the incubation temperatures of 2, 4, 6, and 8 degrees Celsius to observe the germination rates at that temperatures. So we used a spore solution, inoculated them onto the agar plates that were marked with these circles, just so we can find them under the microscope easily. And then we incubated them at initial temperatures, again, 2, 4, 6, and 8 degrees Celsius for the initial temperatures for 6 hours, and then took the plates out, removed them to the incubation temperature of 24 degrees Celsius. After that, we transferred the agar onto the glass slides, put a cover slip on top, and then counted the number of spores that did germinate and did not germinate under the microscope. This is just the data that was recorded by temperature and humidity sensors that went in with the plates into the incubation rooms. And as you can see, the actual temperatures were pretty similar to what we expected for our treatments, which was 2, 4, 6, and 8, although the 6-degree one did malfunction, so it's not pictured here. So the results were that there was an increasing germination percentage with increasing temperatures, but the 6 and 8 degrees Celsius temperatures did not seem to have a significant effect on spore germination, although the 2 and 4 did, but they did not stop germination. So the key findings from this project was that zinc sulfate was an effective chemical defoliant, although it did seem to affect flower numbers, which in turn halved the yield produced by the plant on average. And then hand defoliation would probably be very good for defoliating the plants, but it just wouldn't be commercially viable as it would be very labor intensive and expensive to do. Average fruit weight for individual fruits was not affected by defoliation significantly, although the zinc sulfate ones did vary in size. And then we also have that zinc sulfate treatment did produce fruits that were higher in soluble sugars. And finally, incubation at 2 and 4 degrees for 6 hours did decrease spore germination percentage, although it didn't stop it. So from this project, we now know that defoliation in winter does not affect the fruit yield produced by the plants. We also found out that spore germination was reduced, but it was not completely prevented by temperatures down to 2 degrees Celsius. In further studies, we would need to find out what chemical defoliants would be viable for performing complete defoliation without affecting the yield at all. And incubation temperature ranges can be extended to below 0 degrees to find out this inactivation point of the spore germination. The limitations in this study would probably be that we applied the VBC and protein sprays in conditions that were probably too cold for the sprays to be effective enough, which is why they didn't affect the plants that much. And I would like to just say thank you to everyone listed on this slide, because without them, I would not get to where I'm at in my project right now. And that is all. Thank you.
Very much. That's very interesting. Do we have any questions? Magda, can you see questions online? We can't see. Do we have any questions from anyone in the room? Yep.
Excellent presentation. Thank you. It was really good. So this was a defoliation for 1 year. What are your thoughts on what would happen for subsequent years of the impact of defoliation?
That was also kind of a consideration that we took into mind, because we also kind of hypothesized that maybe because this was only done for 1 year, they might have just used the resources that they already have on reserve. Yeah. But we did find out that the defoliation over the winter period, they did not have a high photosynthetic activity during winter anyway. So that would not affect the yield, I guess, hypothetically. But we wouldn't know until we perform further trials, obviously. But yeah.
Good, thank you.
Right.
Could the defoliation have any other sort of negative effects on blueberry growth, such as are the leaves normally used to sort of harbour species important for the IPM programs for the berries?
Yeah, possibly, I guess. I didn't actually think of that, but I guess it could be a possibility. But we just didn't do any trials to kind of learn about that part. So we wouldn't know, I guess. But it didn't seem to affect the yield per se. Yeah.
Temperature affects the rate of biological processes. Was the temperature effect due to faster germination at higher temperatures? What if the incubation period had been extended, in brackets, on the moist period?
Well, I reckon there would possibly be an even higher germination rate if we extended the incubation temperature at 24 degrees Celsius, because we had a limited time frame that was why we chose the 12-hour time window to kind of incubate it and just see what the effects of the 12-hour incubation at 24 degrees was. But I guess that is a good point. And yes, possibly the germination would probably be higher if they were incubated at 24 degrees for longer.
Thank you. Anyone else? All right. Thank you very much, Nikita. I think we'll let you exit the stage. Thank you. Our next speaker is Siyu Ma, who's here as part of the Western program that some people will know about. It's a joint program that we have with a Southwest University in China. And Siyu has just completed her applied honours. So she's been doing a slightly different program to Nikita. But please come forward, Siyu, and tell us all about land spoilage.
Hello everyone. Thank you for attending my seminar. And the topic of my seminar is exploring the effects of microbial spoilage on sensory quality of land. And my seminar can be divided into 5 parts: introduction, aims, methods, results with discussion, and the conclusion. And first is the introduction part. At first, let me introduce the background of my topic. Plant meat and meat-based products are playing an important role in humans' daily diets because of its high nutritional values. However, high nutritional values of meat also makes it easily spoiled food. The main cause of the spoilage is the growth of the microorganisms. The microorganisms growth on meat can break down the nutrients in meat and produce the metabolites. These metabolites of microorganisms can cause changes in sensory properties because they have their own flavors and color changes. And let me introduce a new technology we used in our experiment. It's gas chromatography. It's a new technology to detect the spoilage metabolites. Briefly speaking, the GC is to use high temperature and temperature changes to break down the chemicals and the components in meat. So we can use this to detect the spoilage metabolites in the meat. We have known that the spoilage process can influence the sensory changes. However, the relationship between the sensory changes and the spoilage haven't been well studied. So that is why we choose it as our topic. By studying the spoilage and the meat sensory, we can understand the shelf life of the meat. And we can also find some methods to detect the spoilage. As we all know, the detection methods of spoilage now have their own limitations, such as time-consuming and high cost. If we can find some new methods, we can solve this situation. So it can help us to have a better control of spoilage, which can help a lot for both consumers and producers. And then is the aims part. So the aim of our experiment is based on these 2 questions. What is the relationship between the microbial information and sensory properties during the spoilage process? And how well does gas chromatography detect spoilage levels? So our aim is to investigate the microbial concentration and sensory changes of lamb packed in modified atmosphere packaging during the spoilage process and test the ability of GC to detect differences in spoilage process. And now let me introduce the methods we used in our experiment. The main materials we use is lamb. Low lamb means in modified atmosphere packaging stored separately at 2 degrees and minus 0.5 degrees for 11 days. These samples were used for future sensory tests and the microbial test. And on each sampling day, 100 grams from each sample treated in vacuum packs and stored in freezer. This is for the future GC test. And 3 replicate sets of samples were made for each temperature and treatment. So the method 1 is sensory detection. 4 experienced lab members were invited to take sensory measurements of the lamp. They set 0 to 8 range of scores as the basis for evaluating the sensory properties of lamp. The lower scores means more unacceptable of the meat. And finally, the odor and the color properties of lamp samples were detected and recorded as the scores. And then is the microbial analysis. 3 10-gram samples from its treatment were placed in a stomach bed containing 90 ml of 0.1% peptide water. And each bed was stomached for 1 minute. 10-fold dilutions of mixed homogeneous was prepared and then was spread on 2 different types of egg plates. The first one is triptych soil egg plates in anaerobic condition. This one is for the total wearable count, and the demand logasa and the sharp egg plates, MIS plates. This one is used for the anaerobic conditions. And all the plates were incubated at 25 degrees for 5 days. Finally, the colonies were counted and calculated as local colony forming units. And method 3 is the GC detection. 1 gram of lamp sample was taken from each sample stored in freezer before, and samples were placed in a stomach bed and added 5 ml ethanol and stomachs for 1 minute. The stomach samples were analyzed in the GC machine. And here are the centimeters we set for the GC oven. 35 degrees for 3 minutes, followed by an increase of 3 degrees per minute to 70 degrees, an increase of 10 degrees per minute to 200 degrees, and an increase of 15 degrees per minute to 215 degrees, and finally holding for 10 minutes. We use nitrogen as a carrier gas, and finally the results were recorded. And here are the results and discussion of the experiments. Here are the sensory results. The left graph is the order changes as the minus 0.5 degree and the 2 degree, and the right graph is the color changes as the minus 0.5 degree and 2 degree. The horizontal coordinates is the time for meat to stay, and the vertical coordinate is the sensory score. We need to know that meat is considered unacceptable when the sensory score reaches 4 and below. And we can see that the decline in sensory properties is almost linear. And the color properties seems to drop faster than the odor at the same temperature, which can be seen that the color change of the lamp is more obvious during the spoilage process compared with odor. And we can also see that the sensory qualities of samples at minus 0.5 degree decrease slower than 2 degree. We think that is because the low temperature can inhibit the microbial spoilage. So the sensory changes of the meats become also slower. And here are the microbial analysis of the experiment. And this graph is the comparison of 2 degree and the minus 0.5 degree on TSA plates, the total wearable count. And the horizontal coordinate is the time for meat to stay, and the vertical coordinate is the number of locked units. Before the analysis, let me talk about some limitation of our experiments. Because the lamp were not well chilled before the transfer to the lab, and the transit time also extended nearly about 3 days. Of course, these factors affected the results of the experiment. We can see that the data of the microbial level is very high during the whole experiment. From the previous study, we can know that the number of local units at around 8 means that it has been very high. So I think this is because when the meat sent to the lab, it hasn't stretched enough as we expected. So we can only see an increase. We can only see an increase in microbial level as the first experiment days, day 0 to day 2. In our expectation, the graph of the microbial should be start at a relatively low number, and then during the time they increase for a short time, and finally gets the max number of the microbial level, and then keep stable. The graph seems that it has reached the state of the stable state. So it is possible that the high starting level resulting in insignificant changes because of the meat. And here are the graphs of the comparison of 2 degree and the -0.5 degree on MIS. It has the similar limitation before the experiment. And again, only the few days of the experiment has a very increased trend at the microbial levels. Compared with the TVC results, there are also some results are in our expectation. We can find that the MIS graph is slightly lower than the TVC graph. That is because the anaerobic environment of the MIS plate inhibited the growth of some microorganisms. The microorganisms produced during the spoilage process, including both aerobic and anaerobic bacteria. And for GC results, the GC experiments are still in progress. We are developing a protocol to finish our measurements. And these are the ongoing work. Samples prepared for GC and find a more suitable method for sample preparation. That is because the GC test has a high quality requirement of the sample. And in conclusion, for sensory changes, color change is significant compared with odor. The lower the temperature and the slower the rate of changes in sensory properties. And for microbial test, the differences were not very significant between the different storage temperature treatment. That is because our experiment has some limitations. The microbial concentration of the lamp was already very high with the experiment performed, so it makes it not very clear for us to analyze. And so I think we need more information to understand the association between the sensory and the microbial level now, because the microbial test is not very clear. And we hope that the GC results will provide more evidence about this relationship. And thank you for these people who helped me a lot. Matt, Jay, Sharing, Laura, Beth, Linda, Edita, and Carly, who helped me a lot during my writing and the experiment part. And the other teachers and students who also have given me a lot of support and help. And here are the references I used in my introduction part. And that's all. Thank you.
I was really hoping to hear the GC results, but I guess I'm going to have to wait and read the thesis when it comes out in a little while. Does anyone have any questions for Siyu? So we have a predominance of agricultural science people in the room and fewer microbiologists. So I I might start while anyone online, if you have questions, please post and we'll get to those in a moment. I know you showed results for the MRS plates and the TBC, and you were talking about the aerobic and the anaerobic.
Yes.
Was there any ideas about which group are more important for spoilage?
Based on the results of my experiments, it's difficult to see which one is important. But during the study, I see some papers before, and I think both anaerobic and aerobic are important because some anaerobic microorganisms, although they are not as much as the aerobic microorganism cause spoilage, but they have better ability to grow in the cold environment. It's based on the package we used in the daily life, the microorganisms can live in a cold environment is more important. So I think most kind of microorganisms we should study.
Is that affected by the modified atmosphere? Does that make a difference? I can't remember, sorry. What the atmosphere in your packaging was. Is that important as well?
Of course, it is also important because compared with vacuum packed meat, the MAP packed meats have a better sensory food and fresh for people to consume. Yes, so it is well used during our daily life. It's really important.
Okay. Is there anyone online? Oh, sorry.
I was just wondering, has works? I know you haven't got the data for the gas chromatography. Has work started? And if so, can you give us an idea about the method or the settings of the GC, how you're going to go about it?
Sorry, do you mean that what's the method used for GC?
The gas chromatography unit. I mean, you're going to undertake some analysis. Are you able to give us some idea of the settings for the machine when you do the analysis, undertake the analysis?
I think at first we should make sure the sample used for GC is clear enough, because the GC machine have a high quality of the sample. If the sample is not clear enough, the GC machine can't give out very accurate results. In fact, I have done some GC tests with editor, and the result is not very expected. That's it, because maybe the method to prepare the sample need to improve, I think.
Thank
you. There's one online from John Bowman. What was the time gap between when the lamb meat was first packed after carcass processing and when you started the experiment?
In fact, for after it passed and sending, I'm not sure. I just know that when it comes to our lab, it has 3 or 4 days later. So I think it influences a lot for the results.
Yes, 3 or 4 days sounds like quite to me. Sorry, that was any other questions?
Just one quick little one. Good job, Siyu. I just, what are your recommendations now that you've done the experiment? What would you say to the family, or who's eating lamb? What temperature would you store your meat? Because for me, I mean, I'm vegetarian, but I'm just would you store it at -0.5? Because then the meat is a bit frozen, or would you store it at 2 degrees? What would you suggest?
In fact, in our daily life, 4 degrees and 2 degrees are the most used use the temperature for storage. However, we have known that the meat is not fresh as expected when it's sent here. So we choose the -0.5 degree to try to find the slower growth of the microorganisms. Although the result is not very good, but we have tried. So we choose -0.5 degree, and 2 degrees is the daily we use temperature.
So you would normally say 2 degrees?
Yes, normally it's 2 degrees. And in our plan, it's also 2 degrees, but we add the -0.5 degree for the second time.
All right, thank you very much, Siyu. So our final speaker this afternoon, Hyemin, has just completed her research thesis in the Masters of Applied Science. So she's our, I think, possibly our final student to go through that program ever. It's very sad, but we will have a new batch of Master's students starting their research thesis next year. So with that, Hyemin, please come and tell us all about your research project.
Okay. Hello, I'm Hyemin. Today I'm going to talk about my project. The topic is yield and profile essential oil Juniper. And this project is cooperated with Thank you so much. So let's start with background. Juniper communis, cold tolerate and mostly distributed in European countries. In the world, the species is largely dioecious. It means they need female male separate flag. And production begins around 6 to 8 years of age and pollinated by wind. And then they produce corn, looks like berries. It takes 2 to 3 years to mature. It's take 2 already. And the mature berry is a key ingredient for gene flavor. In Australia, gene industry predominantly flavored from imported juniper berry from Europe, and the cost is expensive and expect to increase annually. Recently, the common juniper tree has been planted in Australia, and it doesn't start to produce the cones. Compacted berry are traditionally used in distilling industry, and yield and profile essential juniper berry had largely studied in European countries, but Australian industry berry is not fully investigated yet. From this project, we are going to compare yield and profile essential juniper berries grown in different countries, and we'll examine the effect of sample preparation yield and profile by hydrodistillation analysis. And generally, juniper berry is distilled for 2 to 3 hours, but we'll analyze the profile and yield during 2-hour distilling time. And we think essential oil yield and profile will be different with where it is grown because of the environment. And the sample preparation will influence the yield and the profile also. So let's begin with material method. So we were provided small and black or purple berries, but as you can see, there is a red orange color. It's definitely different species. And we got another species from Victoria. These berries were taken monoecious plant. It means that male and female flowers on the same plant. And we just curious where the oil is found. So we cut the berries longitudinally, and then we can obviously see that oil is distributed around the seeds. So, and we got a hint. And the second step is micro-restriction. So why we choose micro-restriction? Because berries, each provided berries was a small amount of to operate hydro distillation except Macedonian berries. So we choose the serpent to use. Serpent was prepared with acetate and ethanol 3:1 ratio, and we added octane as internal standard. And to make finer berry, we used lambda water and pestle. And 2 grams of samples were extracted with 10 years of serpent. So I took this picture after 24 hours, so we can see obviously different colors. It means really different. And the samples filtered with syringe and purified. To analyze the effect of sample preparation, 400 grams of berries were distilled, but the sample was used with intact. And when you're grinding, I use liquid nitrogen. It was take too long, take 40 minutes. And for this one, automated grinding, it just took less than 1 minute, very quick. And we boiled the berries with 2 liters of tap water, and then we got very yellow oil. And to analyze the profile and yield during a 2-hour distillation, we used blended, This is the blended and ground berries. Once the first drop of oil is collected, was observed, we time measured. We measured the time and took the sample every 15 minutes. So as you can see, the first yield was the maximum. To quantitative and qualitative analysis, we used GC and GCMS. So I think the process of GC, she already explained it a lot, I think. And this is our result. But to analyze each piece, there is a solvent, and this one is intact, inner standard. So we decide to overlay all samples. And so we can see some dominant peaks and choose each peaks as number. To identify each peak's name, we got the result just in the morning. So I haven't matched it yet, so it's under process. For this presentation, I will give a number as discussed. And this is our result. So why? Sorry for and this y-axis shows the yield and x-axis shows each origin. So as you can see, the highest yield was from Bulgaria, Italy, and USA as well. And the lowest yield was from Bulgaria. That one is a different species, and Macedonia. And the USA result almost the same with the lowest yield. And this one is a profile. So these axes show each compound, each color depicts each peaks. And the one thing very interesting was different profile between Bulgaria common, Juniperus, and Juniperus. It's always different. And one thing to pick more is Bulgaria, Macedonia, and Juniperus 3 seems too similar. And one thing more to say, Juniperus 1 and 2 are very similar. And this is the sample preparations. So as I showed you before, the blender much better to obtain yield, and it's almost 2 times more than intact berries. And this is the profile. So intact berry has dominant peaks and unknown peaks shared 50-50. And if you see the blender, unknown peaks 25%, but dominant peaks 75%, quite different. And I also prepared the samples there. So after this, we tested the site. So it will be seen in detail. Peak 1 to 4 is less than other samples, but peak 5 was significant. And peak 6 wasn't detected. And if you see the red boxes, the calculated unknown peaks are significant than other 2 samples. This graph shows the yield and profile in 2 hour hydro distillation. So these axes show the composition, and this axis show the yield. And orange line is showing cumulative yield, and that show the interval yield. So as you can see, 15 to 30 minutes, there is a steep increase. And then after that, we can see slow increase. And peak 1 to 4, so 1 to 4 is maximized and then decreased as the distilling time extended. However, peak 5 seems increased during that time, and around 75 minutes, it was maximized. And then they just stable. It was a similar amount of. So, and peak 6 was maximized at 30 minutes, and then they decreases slowly. To sum up, origin is a significant effect on yield and profile. So this would be because the essential oil is synthesized by primary metabolism, but the metabolism is significantly regulated by environment. So it's really very different. And the yield and profile essential oil of various sources will help the industry in making informed decisions. And sample preparation, obviously, ground varies, more efficient to yield and profile. This is because as we ground the berries, the oils can easily break up, released. And 69% of total yield collected at 9 minutes, during a 2-hour hydro distillation. So traditional methods like 2 to 3 hour distillation may not be efficient to produce because its cost increase when you use fossil fuels or something like that. And here is me, my supervisor and business partner. And I'd like to thank you so much for having me here. And thank you so much for helping me a lot, Dr. Elita and Professor Dougal. And thank you, David Losky, to analyze the data. And Dr. Nicholas, who helped me to identify each compound. And thank you, Caroline. She told me how to use the lead distillation, and she told me lots of tips to record. It was very worth. And thank you all the staff and students at TIA.
Excellent, thank you. That's an excellent talk for a Friday afternoon. Don't run off. Do we have any questions from the floor first, and then we'll go online? Michelle.
That was great. I really enjoyed that. Do you know if there are any international standards for juniper oil?
Yes, we have standard in the ISO is like international standard or something. But yeah, I downloaded the file. But we, as you know, we haven't matched each compound. So at the moment, I can say what is what. It's something like this. Just give a number as a P. Yeah, sorry.
There's more results to come. People have to read your thesis to get all the answers. More questions? Yes.
Any idea what the peaks were, the compounds?
Any idea of peaks?
The peaks, there were many peaks that you detected in the GC.
Yes.
The flavor and the compounds are very important.
Yes. So each compound has each smell. So, and it means that it can interpret each compound has chemical properties. So if you have time, we can test it, the smell, and then we can discuss it detail.
I assume these are.
Yes, I prepared.
So we can have a practical demonstration, even if you don't know the names of the compounds yet.
But you can know that.
That's related to what I was going to ask, which was, I noticed that as time went on, the unknown peaks got bigger, which I thought looked potentially quite interesting, but I guess that's something you'll have to explore when you get the rest of your results. Are there any other questions? Yeah. No? No, excellent, thank you. I think we'll let you leave the floor. Thank you to all our students for coming along today and presenting. It is a big effort to get up and talk to all of us, including the people that can't see online. I'd also like to thank the audience for being here and invite you, sorry, online people, but I'd like to invite the people who are here to stay behind for some refreshments and to catch up and ask our presenters a few more of the early questions that you've had time to think about. I'd also like to thank all the staff that have been involved in these projects along with their various industry partners, because without staff support, and that's their supervisors, lab staff, and also the support they get from their friends and family at home, I'm sure you'd agree your projects would be a lot more difficult and not nearly as much fun. Thanks everyone. That's the conclusion of our talks for this semester, and hopefully everyone will be back in 6 months' time to hear the next lot of research seminars from our continuing honours and master's students.